Okay, thank you very much. Thank you very much, everyone. Hope this presentation is interesting. So I'm a postdoc in George's lab. I'm interested in aging and multimorbidity, and this is the work that is part of my PhD, but I've been a postdoc in George's lab also for a while, and this is the work that we are continuing and moving forward. So any suggestions at the end are also very welcome. So okay, so just for a bit of background, we know that aging is very heterogeneous, right? But it's true that there's different aging patterns that exist, or different ageotypes. For example, in this paper, what they found was 4 different patterns in which aging happened based on different organs and how they changed in time, no? And so we know this is happening, and also we know that there's 12 main hallmarks of aging, meaning 12 main molecular pathways that are mainly affected in aging, right? So the hypothesis in our work is basically we want to develop or determine different aging patterns, and then see if these aging patterns are driven by any chance by the failure of specific hallmarks, so specific molecular pathways that are characterized, or a combination of them, right? So this is what we set to do in this work. And so the first thing we did was define these aging patterns ourselves. And for this, we have used UK Biobank, which is a wonderful resource that I'll go a little bit more in detail on what it contains. And the way in which we define these aging patterns is with this biological age marker, which is called phenoage, that you can see here. That is a model that you can see here. It's quite simple, and it's a way in which we can track age in a more objective way than simple chronological values, right? And so the way we compute phenoage is by introducing the values that you see here. So as you can see, they are very standard blood marker information that is widely available. And this phenoage marker has been extensively used in literature. So there's quite some research previously that has worked with this model, but it has been mostly used in a single point in time, no? So you calculate phenoage and then you associate it with health outcomes in a future, but just with this single point in time. But the value of using UK Biobank is that, as you might all well know, but I'm happy to go into details further on. But UK Biobank is this wonderful resource with 500,000 participants from the UK that have been extensively phenotyped. So we have social demographic information, they have been genotyped, they have been imaged, and also something very important that we're taking advantage in this work is that we also have the information on their GP records. So all of the participants are linked to their health records in time, right? And so this is something that we're using because we're calculating phenoage not just in a single point in time, but including these GP records in a longitudinal fashion too. So UK Biobank participants also have had their blood samples taken at different points in time. Well, one for sure, and some others have gotten it twice. So we count with blood marker information from the GP records and from that blood sample data that UK Biobank has taken as part of the study, okay? So what we then continue doing is calculate this phenoage for these participants. But as I said before, we count with the phenoage coming from the UK Biobank source, so from these blood samples that they take as part of the research, which is this big data point that you see here, but also including the phenoage calculated from the GP records blood test data that some participants have. And so in this way, we have these phenoage profiles across time and not just distinguished points in time, okay? And so the hypothesis that we're taking with this phenoage versus age calculation is that we assume that if you have a higher phenoage than your age, so if your predicted phenoage is higher than your age, you are predicted to be unhealthier, right? And if you have a predicted phenoage, so if I'm predicted to be 20 years old when in reality I'm 30, you are predicted to be healthier, right? And so with this hypothesis, we move on forwards. And what we continue doing is fitting a linear model into each of these data points. And in this way, what we end up is with 4 trajectories of phenoage, right? We end up with participants that go from this unhealthier kind of phenotype, right? That were constantly in this unhealthier kind of scenario into a healthier one, participants that are constantly in a healthy trajectory. So their data points always were found there. Participants that went from an unhealthy trajectory to an unhealthier one, and participants that remain constantly in this unhealthier side, right? So we have defined our aging patterns in this way. And what we then continue to do is try and characterize them as best as we can. And we can do so, we can do it quite extensively because UK Biobank has all of these very interesting descriptors of these participants too. So we start describing them, looking at lifestyle information and key health outcomes. We then continue describing these different trajectories in terms of their disease makeup, because we have access to hospital inpatient data of these participants. So we have all the ICD-10 codes that have been coded for these participants in time. And what we do is add an extra layer to these ICD-10 codes and just map these ICD-10 codes to see chronic disease categories, because we know that age is the main risk factor for the development of chronic diseases. So we are just, in order to have a more kind of structured analysis, we're just focused on chronic diseases at this point, okay? So we are focusing on these 60 chronic disease categories that have been mapped using the paper that I referenced here. And then we also try and characterize these participants genetically, because we also have them information, the genotypic information, right? And so, yes, all of this information is available in UK Biobank. And so very briefly, just for you to see, in terms of health outcomes and lifestyle information, this is the type of data that we looked at, right? So here we have the 4 clusters, or the 4 aging trajectories that I mentioned before. And we looked at very standard health outcome data, such as deprivation index, health rating, BMI, smoking, activity, comorbidities, etc. And what we see like continuously is that the healthiest kind of phenotype, the one that always remained in the healthy trajectory, has better health outcomes than the unhealthiest one. And for example, as an example, you can see that mortality-wise, we only have a 2% in the healthiest one, whereas the most unhealthiest, the one that always remained unhealthy, has an 11.8%. And this is constant within all of these different lifestyle factors. Also, kind of interesting was also that we found more females. That is always something constantly seen in this kind of studies, more females in the healthiest one and more males in the unhealthiest one, okay? So that was just a brief overview of what the different aging patterns looked like. And then we went into looking at disease makeup, right? And here is for you to see a bit more what these 60 disease categories that I mentioned before look like. So in the end, we had these ICD-10 codes mapped into these 60 categories, such as that can be summarized into hypertension, colitis, dyslipidemia, etc. And summarizing what we found there was that there was a cardiometabolic disease enrichment in the unhealthiest phenotype. So for example, you can see here diabetes in the whole cohort had a prevalence of 10%, but in the unhealthiest one, it went all the way up to 26%, whereas in the healthiest one, we had only 2%. And the same thing happened with more cardiac-related diseases. And then what we looked a little bit more too is into what the interaction of these diseases was. So from here, we calculated the relative risk ratio of disease pairs. And very briefly, what you can see in this image is that the healthiest one had less number of diseases and they were less connected between them, meaning that they were less multimorbid. And the unhealthiest one, the opposite, there were more diseases present and they were more connected between them because the patients were more multimorbid too. Also, something to mention is that the age was quite similar between all clusters. And then lastly, we looked into the genetic makeup. So something interesting was that we performed GWAS in each of the 4 different categories. And what we saw is that there was only genetic associations, significant genetic associations found in those extreme phenotypes that I mentioned. So the healthy that always remained healthy and the one that always remained unhealthy. In the ones that changed categories, so went from the healthier phenotype to the unhealthy, or went from the unhealthy to the healthy, no significant associations were found. And so we went into characterizing a little bit better these remaining healthy SNPs. And what we found, we annotated the SNPs using QTL data and proximity. And what we found is that the leading SNP, for example, in the remaining healthy is this ApoE4 variant, well known in aging, highly associated to aging. And we also found 2 SNPs associated with longevity, which was quite interesting. And we proceeded after the annotation with functional enrichment. And in very general terms, what we found is that there was an immune signature driving these remaining healthier phenotypes, whereas the unhealthy remaining unhealthy was mainly driven by metabolic signature and lipid biosynthesis. And we want to highlight that what we found is that only transcription factor targets were found in these unhealthier kind of phenotype, and we want to highlight these two, which are steroid regulatory element binding transcription factor 1 and 2, that have been previously highlighted as being key controllers of the molecular footprint of aging, and are highly associated to at least 3, I here put 2, but at least 3 molecular hallmarks that I mentioned before that have been associated with aging: chronic inflammation, mitochondrial dysfunction, and autophagy. So, well, just as a conclusion, so we have defined our own aging patterns, right? And we have characterized this as extensively as we could, thanks to UK Biobank that has all of this information. And then for the future, what we are really interested in doing, and for example, there was a talk here a couple of weeks ago on multimorbidity and trajectories and sequence of diseases in time. That's something that we are really interested in pursuing. So looking a little bit more in detail, what is the disease makeup of these participants in each trajectory, not only in a pairwise association manner, but also in sequence, because we have the diagnosis in time. So what we would be interested in seeing is how the trajectories of diseases change between the different patterns. If we see specific clusters of diseases found in clusters in one of the trajectories more than the other, how can we associate this with genetic makeup? And then, of course, the idea is to see if there's any underlying shared molecular mechanisms between these trajectories and between the development of these diseases. Basically, because in the end, this idea like multimorbidity increases with age in the same way that diseases increase with age. And it's true that some hallmarks have been linked to the development of diseases. For example, chronic inflammation is highly associated to the development of most chronic diseases, right? And so the idea is like, could we highlight specific hallmarks underlying cluster of diseases, not only in our aging patterns, but in these specific clusters that might appear. So that's it. Thank you very much to my lab, which is the Cocutus lab here at Institute of Translational Medicine, my supervisors, and this is the work that I will continue doing. So any suggestions are very welcome. Thank you very much.